Hello, everyone. Welcome to this webinar. I really want to thank everyone for attending, for joining us. We will start in a few minutes so that we can allow everyone to connect, to activate the, the captions and the translation. I hope you were able to see how to do it. Just in case, I will make a quick review about how to do it. Okay. If someone has not activated the translations yet, you may find this useful. Okay. So I will share my screen. You have a video that one of my colleagues made, which is much better than my translation. But you will see that my configuration is in Spanish. So just follow my lead, let's say, so that you can make the translation as well. Okay, so here you have this menu. Okay, these three buttons. So you just press here, you go a little down, configuration, yeah, it will open here. Even though it's in Spanish, you can see it's very similar to English. Subtitles, okay, and here translated subtitles. And here you open this drop down menu and you can choose your language. As soon as you activate it, you will see, if I continue talking, you will have the translation simultaneous. Hmm? While I talk, you should find the translations automatically in your screen. Okay, I will just keep with the no subtitles, okay, but I hope you are able to use it. Okay, so let's start. So before, okay, I will start with a little introduction about myself and the team that is joining together with me, assisting me in this webinar. So my name is Antonella Sdrigotti. I've been working at Terragene for 13 years. I nowadays I lead the team of product specialists and clinical educators for basically the world, uh, all countries, but Latin America, which is led by other team. And I will be giving the presentation myself. And together with me is here Carolina Mazzucchelli. She works in my team as well, and she will be helping me if someone raises their hand or asks something in the chat box. She will help me with this webinar. Thank you very
Well,
much, Caro, for joining.
welcome. Hello, everyone. Hello, and thank you for joining us.
Yes. And also here are other members of the marketing team. Between them is Andres also. So thank you very much. The marketing team is also helping us with assisting us just in case with the technical part of the webinar. So thank you very much because this is a teamwork and we are very happy to work together to bring you a good presentation. So, without further introduction, and once again, thanking everyone joining us in this webinar. Good afternoon or good night for those that are far away from Argentina. Here is 8:35 in the morning. So I hope everyone is enjoying the afternoon and this presentation. Of course, when I finish, it will take around 1 hour, 1 hour and 10 minutes. Okay. But as soon as I finish, we will give time for you to make questions. You can write them down in the chat box. Okay. Or if someone wants to open the mic, we can also listen to your questions as well. Okay. So I will share my screen now once again, now to share the presentation. Caro, just check. Good.
I was muted. Yes. Yes, I'm
Excellent.
done.
Excellent.
Excellent.
Okay. So I hope most of the people that registered for the webinar are already in. So I will start with the presentation. So as you have all seen, the title of this presentation is Ensuring Effective Cleaning, Advanced Monitoring Solutions for CSSDs. And I will lead the presentation together with Carol that will assist me in case you have any questions. Okay, so let's start. So why should cleaning, monitoring, verification, be routine in CSSDs? Because we sterilize what we believe is clean, but how often do we prove it? So sterilization failure rarely starts in the sterilizer. It starts in the washer. Cleaning is not a visual process. It is a measurable biochemical process. If organic residues remain, sterilization becomes a gamble. So this is the illusion of the gleaming tray. What we see, what remains, what really remains. Under intense surgical lights, an instrument tray looks absolutely perfect, but true cleanliness exists on a scale completely invisible to the naked eye. So what happens? Incomplete washing leave body fluids and tissues on the instruments, which will provoke a protein shedding. So microorganisms hide beneath these invisible protective proteins. This will mean that sterilization will be blocked. High-level disinfection and sterilization agents cannot penetrate this protein barrier. And as a consequence, this will result on patient risk because contaminated instruments reach the operating room. So without proper cleaning, disinfection and sterilization can fail. So why are organic residues a direct hazard? Because residual soils provide protective niches for bacteria, supplies nutrients for microbial growth, interfere with the antimicrobial activity of the disinfectants, and support biofilm formation. And we all know here that biofilm is the most significant threat in the CSSD. This means that effective washing will give, as a result, safe patients because washing is the first critical step in the medical instrument reprocessing, and without it, everything else fails. But let's start with definitions and objectives because when we interchange and we mix these definitions or these uh, words, we can make wrong decisions about how to monitor them. Okay, so let's start with washing. Washing is a physical action, which objective is removal of organic and inorganic matter from reusable instruments after use. On the other hand, we have disinfection, which is a biochemical action. And the objective of disinfection is inactivation or killing of microorganisms or any other type of living cell from the reusable instruments after washing. What about cleaning? By definition, cleaning is remove of dirt, just like washing. But in the hospital practice, we use it as a synonym of hygiene, which is washing plus disinfection. On the other hand, but not least important, hygiene. by definition, is a set of practices to prevent contamination risks and protect health. In the hospital practice, it is washing plus disinfection, and we use it as a synonym of cleaning. This is very important because we, on the daily practice, we interchange these words, and scientifically, they are not synonyms, and confusing them leads to incorrect monitoring strategies. So, why to distinguish washing from disinfection monitoring and verification? Because washing and disinfection are sequential but biologically different processes. The success of disinfection depends on the success of washing, and monitoring strategies must reflect this difference. So we will review the science behind this and also the solutions that Terragen gives to monitor all these different processes. So which are the principles involved in monitoring and verification of a cleaning process? We have to establish SOPs or standard operating practices or procedures based on published recommended practices or guidelines, validation and process monitoring, and measuring and evaluating residual contaminant after the cleaning process, meaning cleaning verification or hygiene verification. We will see the two of them. So let's start with establishing SOPs based on published recommended practices or guidelines. We see here just,
Sorry, Anto,
yeah,
somebody raised a hand. I don't know, Camelia.
if there is a question, I kindly ask you for finishing the presentation. And
Okay,
after we
at the
finish,
end. Perfect.
yeah,
Perfect.
after
Yeah.
we finish, sorry, I forgot to say that. When we finish, or maybe I said, I don't know, when we finish the presentation, we will give some time to ask for questions, okay? By chat box or opening the mic. Yeah. So these are just examples of the practices or guidelines in different regions. Of course, we also have the local standard or regulations, but these are regional regulations that most countries tend to choose from one or the other. These are just examples of the most popular that we see worldwide. And here the examples are the Health Technical Memorandum from the UK, the APSIC or Asia Pacific Society of Infection Control, the ISO 15883, and ANSI AMI ST 98 about specifically about washing and cleaning monitoring, and the ST 79 that we all have heard of, which is from reprocessing instruments in the CSSD in general. Okay, so these are just some examples of them where you can find, if you want to choose one or the other, you can find SOPs that you can follow and adapt in the CSSD. Okay, but all of them have a coincidence in terms of validation. Okay, so when we are talking about washers, I mean, this is also true for autoclaves or other type of sterilizers, but specifically focusing on washing processes and thermal disinfection, we have to perform a validation. Validation is a documented program to demonstrate that the process constantly delivers products that match the specifications. And the validation process has three steps. We have the installation qualification, operational qualification, and performance qualification. The installation qualification is where we have to ask, is the equipment provided and installed to specifications? Is the new equipment installed as it's supposed to be installed and working and I have expected to work? Then we have the operational qualification. Does it operate correctly within the limits? And this is with an empty chamber. Okay, so we have to make sure that it operates correspondingly, but without the load. And last but not least important, we have the performance qualification, which is, does it clean and disinfect specific loads? So this is where I have to validate each of my instruments, or at least the most challenging instruments, and perform specific loads to demonstrate that this device is working correctly and cleaning correctly my load. Okay. And we also have what we call routine control. Okay, this is validation. Normally, and in most countries, is once per year, at least the performance qualification and the operational qualification. But we also have routine control. And in routine control, we have two different aspects of routine control. We have monitoring and we have verification. Caro, just in case, just check with Andres, just in case they are raising the hand, because if there's some technical issues,
No,
maybe.
no,
No? Okay,
no,
okay,
no. It's fine, Anto.
okay, okay. I'm worried that something is not being correctly delivered, but if you confirm, that's
No,
fine.
there is
Yeah.
no hands raised and no questions in the chat.
Okay. Great. So, monitoring. What is monitoring? Is a continual checking, supervising, critically observing, or determining the status in order to identify change from the performance required or expected, performance level required or expected. This means focus on the activity, continuous oversight of process parameters. Okay, this is what we will do on a daily basis. And what is a verification? Is the confirmation through the provision of objective evidence that specified requirements have been fulfilled. This means focus is on the outcome. We confirm that the result meets a predetermined specification. And we will see what Terragin offers for monitoring and for verification. Okay, so we will start with the washing monitoring solutions. About washing, which are the parameters that influence washing? So if we are going to monitor, we are going to focus on meeting the parameters. Which are these parameters in a washing process inside the washer? These are the five main parameters that influence the outcome of a washing process. In the center, water. When we talk about water, we are talking about the quality of the water. Is not every water, even if we say water, is the same. We have to pre-treat the water that we are going to use in the washer. Some regions, some countries still don't use pre-treated water during the full cycle. The thing about this, and it's only mandatory to use a pre-treated water when we are going to make the last rinse before drying. But if we use pre-treated water during the full cycle, we make sure that we preserve the instrument, we preserve the machine, and we preserve the performance of the detergent that we are using, because everything that is dissolved in the water will influence all of this. So preservation of instruments, washer, and performance of the detergent is guaranteed. When we use water, we know the quality. What what is the quality of that water we are using, in order to make sure that this process will be successful over time also. Then we have chemistry. Chemistry and temperature. Chemistry and temperature work together, because we have to use a temperature during the washing phase according to the recommendations of temperature of the manufacturer of the detergent. The efficiency of the detergent is very important in terms of temperature. If we don't use the right temperature, we can decrease the efficiency of that detergent. Okay. Of course, we have to choose between enzymatic or alkaline. Okay, and use the right dose. Using a higher concentration not always will mean a better performance. Okay, we have to use the dose that is recommended by the manufacturer. Then we also have mechanics. Mechanics is one of the most important aspects because it's how the washer is working. And it's very difficult to confirm that because when we start the cycle and all the water sprays start to work, it's very difficult to check if all the water sprays are working correctly. And this, in the end, will influence, according to the location of a specific instrument, will be uh, good or bad for the instrument if a water spray is clogged, is not working. Okay? Also with the rotating arms. Sometimes it's easy to see that a rotating arm has stopped. But what happens if it stops rotating? Those instruments will not be impinged by the water spray correctly. So it will mean that in that location, the instruments may not be correctly cleaned. Okay? So and also the pressure of the water spray. So all these mechanics, performance of the washer needs to be monitored. And and it's important parameter. And of course, last but not least, the time. We have to validate the time we are going to spend to clean different type of loads because not all of them are clean with the same difficulty. Okay. And also, which is not here, but one other parameter that influences the outcome is the load. Do not overload the chamber because we are in a hurry, because that will be very detrimental for the efficiency of that washing. So it's very important to keep all the instruments open, disarmed, as according to the manufacturers, for the manufacturer of the medical device, and right open to the water spray, to the washing process. So what is the solution that we offer to monitor all these parameters? These parameters will integrate, they are not independent. So the success in the end is a very thin fine tuning between them that will make sure that all the instruments will be correctly washed. A very simple indicator to monitor this type of parameters is the indicators you see here on the screen. ChemDICE plot, we call the line, and the codes are CDW-A3 and A4. So these are washing indicators. They are synthetic substrate, thermostable. This means it's like a plastic, it's not paper-based. And they are printed with a test soil of organic components, proteins, carbohydrates, fatty acids, and colorants, all natural components. And this composition of this test soil, which is of course according to the ISO standard, residues, organic residues, as well as the difficulty or the adherence of these human residues over the surfaces of instruments. Okay, so in the end, this test soil simulates what happens with the real human residues that can remain over the instruments. Okay, we have two different type of challenges because there are many manufacturers worldwide that work with different parameters. Most of the validation processes and the intended use of washers are very strict and very high. So this is why we always recommend to use the high challenge indicator, the pink one. But also some items need to be more mildly reprocessed and using milder detergents, lower temperatures, shorter times. So this is why we offer also a second, less challenging but still challenging test soil, which is the CDWA3, that will work in these less strong, let's say, processes. But in the end, both of them have these advantages that you see on the right side of the screen, which is they don't have any blood components. All the formulation we use is organic, and it has automatic interpretation. This means also automatic registration, okay? Because these indicators can be read and interpreted with our technologies, which is Transantolanes and Bionova Q application. These are cell phone applications that use your camera to take a picture of the indicator and evaluate if it's a pass or a failure. We will see how to interpret, okay? But these indicators in the end can be automatically read interpreted, and digitalized. So you can keep a digital record of the result in our software and trace down everything about every part of the process. Okay, of course, it's very easy to use. It's a very accessible cost as compared with other brands that have more integrated indicators that make them more costly. And this means that if we have a more accessible indicator, we can monitor more frequently and not choosing which cycle I will monitor. The idea is to monitor as most many cycles possible, preferably every cycle. Okay. And of course, I did not mention this, but they can be used both for load release or monitoring, process monitoring, as well as for the validation of the machine. So these are the automatic traceability that I have mentioned. Okay, two different softwares, two different applications for the cell phone, but the same artificial intelligence used for the interpretation and digitalization. Both of these indicators, even though the pictures show the pink one, the blue one is exactly the same use, but both of them can be used both for thermal disinfection as well as ultrasonic cleaners. We have two different holders for this, okay, because ultrasonic washers work in a different way through cavitation. We will see that later. So we have to use the indicator in this holder, which does not have a front part to protect the indicator because everything will be submerged. On the other hand, and both of them have a clip at the back so that you can secure the indicator on the basket that you are going to use for monitoring. On the thermal disinfection part, we see that actually we have like a configuration on top to protect the indicator. Because what we want to do, we want to simulate what actually happens with the instruments inside the chamber. Not all items will be impinged or be reached by the water spray. So we want to check that this is true also for the indicator. And if the indicator can be correctly washed out, it means that those instruments that have the same situation will also be cleaned. Okay? So depending on the results, we can design a corrective action. This will be the initial, as you can see here also. The accepted is a total removal of the test soil. And depending on the residues remaining on the indicators, I can decide or have decision about chemical or physical problems and make corrective actions. We have to cover too many indicators. I will go quickly with this one, but in case you have questions, we can review that later. We also have ultrasonic cleaning, and this machine has a little difference. As you all know, the ultrasonic washer is used for cleaning um, and removing soil from areas difficult to clean by other methods, such as joints, crevices, lumens, blind holes, and complex geometries. And the particularity about ultrasonic washing is that the microscopic scrubbing or cleaning is thanks to cavitation. Mm? So besides all the parameters that we have already seen for the regular washer machine, we also have frequency, which is a very important factor. And in here also, water quality, chemistry, temperature are very important because here there is a fine tuning that will define the cavitation energy, which is in the end the one that provokes the removal of the organic residues from the instruments. Frequency is very important because actually the machine needs to work in the ultrasonic range. And in this ultrasonic range, most of the ultrasonic washers work between 35 to 45 kilohertz. Temperature also cannot be too low or too high, because this will provoke a difference in the cavitation energy, and the chemistry also, because everything dissolved in the water will affect the cavitation energy. And why is very important to monitor the cavitation energy? Because the cavitation will not work just because the machine is running. Mm? Every day, and I would say every time we change the solution, the parameters or variables can change and the energy distribution can be different. So as I have mentioned, if the temperature is too high, they will provoke vapor pressure that will cushion the cavitation energy or the implosion of these microscopic bubbles that remove the organic residues, provoking dead zones. Everyone that has used an ultrasonic washer knows that we have to degas the solution. What is degasification? Is elimination of the gases dissolved in the water. And these are the bubbles that we can actually see when we start the machine. These bubbles need to go out. These are air bubbles trapped in the water that decrease a lot the cavitation energy. So this is why we make the gasification before we put the load. Mm? We eliminate this air inside the water. Of course, the load and the frequency. Mm? Heavy loads or incorrect frequencies will distort the waves, the energy of cavitation. And basket design. We have to use the appropriate baskets, the ones that come with the machine or are recommended by the manufacturer in terms of materials. We have to make sure that we are not decreasing the cavitation energy due to using tight meshes or plastic baskets. So consistent performance requires verified practices, not assumptions. And this is the reason why we have this ultrasonic cavitation performance indicator. Mm? This indicator, ChemDye CDWU or CDWZ, are indicators. Both of them are very similar. There's a very little difference. What you see in the screen is the version Z. Okay? This one is a glass ampoule with the blue reactive solution with ceramic cylinders immersed in it. Initial color is blue. The yellow you see also is the final color, but the initial color is blue. The CDWU is exactly the same, but instead of having ceramic cylinders inside, it has glass beds or pearls. Okay, it's just a material and format difference due to patent reasons, which no longer exist. So both of them are completely the same and can be perfectly used everywhere. So what is the aim of this indicator? What will happen if we put this indicator inside the machine once I have the solution, the detergent, and the right temperature? And we start after the gasification, we make a short cycle. The vibration of the cylinders in this case will trigger a reaction, a chemical reaction that will start a color change from this blue through different shades of green until arriving to the final. Let me start a pointer. Um, Here, okay? Through different shades of green until arriving to the correct cavitation energy, which is this yellow, okay? We also have specific hooks or holder to secure the indicator inside the washer. Thanks to this shape, you can put it on the basket and make sure that the indicator will not move around due to the vibration, okay? And that you are going actually to monitor the performance of the transducer in that location, okay? What is the aim of this indicator? To validate the performance of the washer, ultrasonic washer, that the washer regardless of the size and the number of transducers that are provoking this ultrasonic vibration and provoking the cavitation inside water, detergent, temperature, and all the parameters that influence, we make sure that at the location we are testing the energy is delivered correctly. And this will mean that any instrument in that location will be correctly cleaned. If we are making a periodical test, this will use more indicators, but will be less frequent. If we use it for routine, every time we change the solution, we have a new situation, new water, new temperature, new detergent. So we should test it again with less amount of indicators because of course it's just a routine. Just to make sure that in different locations, the cavitation energy is right. Okay, so what are the challenges of using this? Sorry, I will go back just a little bit. Just for those that is the first time they see this indicator, I always like to say something that I think is very easy to understand. What is the use? This indicator, I like to say, is the Bowie-Dick test of the ultrasonic washer. Okay, so Bowie-Dick test, we have to make one every day before we start with the load. Okay, before we put the load, we have to preheat the chamber, in this case, the gasification, and everything is set. I just do a Bowie test. If everything's okay, I can put the load and continue with the work. This is the same, okay, with the washing solution prepared at the right temperature and the gasification. I just put a few of them in different locations. I run them. If everything's okay, I can put the load and I can use one of the washing indicators with a corresponding holder to release the load. Okay? I think this simplifies what is the aim of this indicator. What are the challenges of using this type of indicators? The subjective interpretation. Visual reading depends on individual judgment, leading to variability and inconsistent evaluations of cavitation energy. Operational uncertainty. Without standardized criteria, staff may hesitate when making decisions about process performance and corrective actions. And lack of traceability, because visual results do not generate documented records. Once the indicator is thrown away, the proof is gone, and this indicator cannot be kept and documented because it will revert the color. The use is in the moment, and the shape does not make it easy to be kept. Okay, so lack of traceability. So this critical flaw limits trend analysis, audit support, and long-term process monitoring required in modern facilities nowadays. So what is our solution? And this is a new product, Bionova CaviTest, which is in the end an automatic reader of the cavitation indicators. Okay, so this automatic reader standardizes the interpretation of both these indicators, delivering objective reading, quantifiable cavitation index, clear pass-fail results, documented traceability, and standardized decision criteria. As you can see in the screen, it has 3 positions. This is an automatic readout. There is no incubation. You just place the indicator in one position, press the corresponding button of that position, and automatically the auto reader will give you a result. Of course, as all our electronic devices, these are connected with both our softwares, Bionova Cloud and Bionova Q, for automatic traceability and digitalization of the results. We have an inbuilt printer that will automatically print a result with the cavitation index and the pass-fail result. How it works. This machine, this auto reader, detects the final color of the indicators through precise light transmittant measurement. And these RGB or red, green, blue values are mathematically converted into HSV parameters, hue, saturation value, or brightness parameters. And we establish a correlation That is that converts these values into a cavitation index that goes from 0 from 130, and the index, when the index is equal or more than 100, is a yellow color, a pass result. And when this cavitation index is less than 100, there's a color deviation that could go from different shades of light green that confuses to yellow until 0, which is actually the blue initial color. Okay? As you can see, the automatic reader will automatically print the ticket with the information you can see in the screen. Two examples of a pass and a failure. You will have the batch and serial number of the machine, the ticket number for traceability also between the printed ticket and the digitalization, the program being used, the position being used, the date and time, the result if it's a pass or a failure, and the index. Mm. So you can see we have an instant reading, it's very easy to use, reliable results, printed evidence, and standardized process control, thus solving the previous challenges of the interpretation, just relying on visual interpretation. So let's move on. We have only talked about washing so far, but what about the thermal disinfection that happens inside most washer disinfectors nowadays? So what is a thermal disinfection? Is the inactivation of microorganisms with water at a high temperature for a certain amount of time. So which are the parameters? Time and temperature. That's it. Okay? The disinfection effects, or these two parameters, are defined through the A0 value, which is also defined in the ISO 1583 Part 1 standard. The A0 value will denote the level of inactivation. So this is just the parameter we need to know to understand the level of disinfection we reach with our washer disinfector. Okay, it does not just matter the time and the temperature, because different manufacturers can work with different combination, but in the end different combinations of time and temperature can reach the same A0 value. And this is what I try to show you on this table. Here you have temperature and a certain A0. Just two examples of the most A0 values used in the market. This 600 is the minimum one for critical devices, for critical instruments. And highlighted in green, you have the mostly used. So 90 degrees nowadays, or 93, but 90 is most worldwide use, a higher percentage of the machines. And in just 1 minute, I can reach the minimum level of disinfection, the ISO standard, um, and the classification of the medical devices intends to have when performing a thermal disinfection inside a washing machine. Just as another example, if my machine is not working at 90, it's working at 80, you see I need 10 minutes, but I will reach the same level of disinfection, of inactivation of microorganisms and cells. Okay? The same with a zero value, 3000, which nowadays is mostly used because it adds just an extra guarantee, an extra safety of disinfection just in 5 minutes at 90 degrees. So just showing you the most regular or most used combination of parameters for thermal disinfection nowadays. Okay, the higher the zero value, the more microorganisms I'm able to inactivate, or the most resistant microorganisms I can inactivate. Okay, so what is the solution we give for this specific step? Because it has no relationship at all with washing. Here we expect to kill or inactivate microorganisms, not to remove them. Remove them is washing. So here we want to corroborate the combination of time and temperature to guarantee the A0 value or level of disinfection. So this is the IT27W line of thermal disinfection indicators. They are designed to react to moist heat disinfection processes inside washer disinfectors. They assure the efficiency or the level of a zero value or of disinfection. And we have a green ink that turns to purple when time and temperature are correctly integrated to reach the A0 value. As you can see here, the unprocessed in the color is this one, and the failures will be seen in different shades of color between the initial and the reference. You see here, this is the reference one. You have it printed on your indicator so that you don't have any hesitation about which is the final color. If it's darker, it means that you just over-exceeded the minimum to guarantee disinfection. So if it's darker than the reference, is better. And of course, as you can see here, it can be used with the same holder as we use for the washer indicator. So two different holders, right? One for the washer indicator and the other for the thermal disinfection indicator. This way, we make sure we monitor both different steps of a washer disinfection process. These are the three options we offer for the three most used A0 values in the world. IT27W1, here just examples of 1 minute and 90 degrees because it's the most used. A0 600, inactivation of bacteria, fungi, and heat-sensitive viruses, just in 1 minute. IT27W5, A0 value 3000, generally 5 minutes at 90 degrees. And this one has efficacy against heat-resistant viruses like hepatitis. And IT27W10, which is for the epidemic program. Mm, The A0 value here says more than 10,000 is around 12,000 and is reached in 10 minutes at 93 degrees. This type of program is used when we have an outbreak and we don't know the microorganism that is generating this outbreak. So just in case, we add an extra disinfection guarantee to make sure that regardless of the origin of this microorganism, we will inactivate it. Okay? This is why we offer these three options for the three mostly used combinations of time and temperature to reach these specific A0 values. So in the end, as I have anticipated, washer disinfector monitoring will be perfectly controlled or monitored when we have two different indicators for monitoring two different steps. A physical removal of organic residues with a specific indicator that will show this elimination or removal of organic residues, and the thermal disinfection um, step that will make sure that I'm having sorry, and the thermal disinfection indicator that will specifically monitor the of time and temperature that makes sure that I will reach the right disinfection level or inactivation of microorganisms. Two different processes happening inside one full cycle. By using different indicators, we will make sure that we have both of them successfully reached. In this case, I'm showing you different results that we can reach, where we have in the first row a successful washing step and a successful thermodisinfection step. Mm? Both of them okay. In this one, the second row, we have a successful washing step, but something happened with the time or the temperature in that location where the indicator was positioned that is not making me to guarantee the right level of disinfection I was expecting. Meaning I cannot guarantee the right level of disinfection of the instruments in that location. And this is why it's so important, because inside the washing machine, the sensors are not located where the load is. They are located in specific parts of the washer machine, but we have to make sure that the right temperature reaches the instruments. And this is why we need this type of indicators, as well as the washing indicators. Third row, here we can see we have a failure in the washing step, but the thermal disinfection step was right. And the last one is a total failure in both steps. So you can see that in the end, if we don't have separated monitoring for these two, we will be missing the thermal disinfection efficiency test. Okay, so we have finished the monitoring because we were talking here about the activity. We're monitoring the processes, the parameter processes. But now we are going to verify. And how we are going to verify? By measuring and evaluating residual contaminants after the cleaning process. Traditionally, and we will not do this on a daily or, a cycle basis, okay? So how we make sure that actually the result is a total clean instrument? We have to test the instrument, right? So how this was performed traditionally by visual examination under the scopes and or the magnifying lamps, sorry, and borescopes cameras mm, for internal channels. But the current trend is to go beyond the visible soil, beyond our only the visual check. How? By verification methods that allow the assessment of specific analytes of both external and internal surfaces. How? Establishing reliable markers and tests. And what about markers and tests? And here is the big question that is controversial, but is still a problem in the CSSD. Which one is more appropriate, ATP or protein for instrument hygiene monitoring inside the CSSD? Hygiene monitoring or cleaning monitoring? We are going to answer that question. ATP testing was born for food industry, but was misplaced in the CSSD. We will see why. In the food industry, ATP was created to detect microorganisms after cleaning or disinfection of the machinery surfaces, not instrument, medical devices. But the hospitals adopted ATP, reacting to healthcare-associated infection outbreaks. At that time, no other marker or test existed. ATP was a solution at that time, but because there was no better option. But this is mistakenly used in the CSSD nowadays, that other options have been investigated, scientifically demonstrated, and developed products according to this need. So today, ATP is not the best tool for cleaning verification in CSSD. Protein testing is. And we will see a list of reasons why. Why protein testing matters? Blood and human tissues contain mainly proteins. Proteins perfectly represent the main constituents of human tissues derived contaminations. Proteins are the main constituents of microorganism cells and the strongest scaffolds in biofilms. So proteins perfectly represent the main constituents of microbiological derived contaminations. Proteins, in the end, human or microorganisms, proteins represent a direct measure of organic and microbial contamination. Proteins are the most difficult to remove residues during a cleaning process, so perfectly represent the highest challenge for cleaning processes. A dead cell will lose the ATP content. but proteins will remain, meaning there is still a contamination in the instrument. If I kill a microorganism, but that dead microorganism remains on the surface of the instrument, the contamination will still be there because proteins will remain and biofilm can form and protection from disinfectants and sterilization will remain. So this is a contamination. So proteins directly represent organic contamination, while ATP shows metabolic activity, meaning only live cells. Not all microorganisms contain ATP. Viruses do not contain ATP, and prions are proteins. That's all the structure they have. So proteins perfectly represent the risk of total potential contamination, regardless of the origin of the cell or microorganism. And last but not least, with the protein test, we reduce and monitor the prion contamination risk because proteins is the only molecule that allows to assess and monitor the risk of prion contamination. This is so true that in the last, let's say, 5 years, most standards have updated. Those that have already considered this are not updated, like the HTM that for long years have already set up the protein test as mandatory. But most of the more worldwide recommendations, like ISO, which is a standard, or AAMI guidelines, have updated and reconsidered protein test and quantification of proteins in their recommendations. No mandatory, but it is a recommendation because what I intend to show here, just with the same SOPs that I have shown at the beginning for validation and, and monitoring, for verification, there is also a trend in suggesting in the concern of protein testing to verify that actually my washing process is removing organic residues from the instruments and not only setting up or monitoring the parameters, but monitoring the outcome, which is protein-free instruments. Mm? So you can see here just a summary, because I will not go through the specific values. This is not the intention, but it is important to see how the standards or guidelines are moving towards protein quantification in order to verify the cleanliness of instruments and the effectiveness of washing processes. So let's see which is the cleaning verification test that Terragen offers, which is the only real quantitative in the market. For surfaces, we have ChemDye Pro 1 Micro. I think most people here already know this product, okay, but it's always good to refresh this concept. As you can see here, the protein test is basically a protein swab within a device that looks like a pen. On one hand, we have the swab, which is the one that obviously will take the sample from the instrument. And on the other hand, we have the pen or the device that will harbor the reactive solution. As you can see on the right side, the reactive solution initial color is green. So when I activate the pen, which is actually mixing two solutions to make the right reactive solution green, this solution will react with the proteins present in the sample and will change to purple. The darker the purple, the more proteins I have on my instrument. This product, since we are saying is a quantitative device, product or test, we need an incubator calibrated to make the quantification. This is the reason why you see on the screen the Mini Pro, which is a device specifically developed and dedicated for protein tests with 4 minutes incubation. As in the same way as all our devices, You will also have a printed ticket because it has a thermal printer built in on the device, and also can be connected with our softwares for digitalization and traceability of the results. And most importantly, trend analysis of protein tests and washer performance. Also, other devices such as IC1020FR and IC1020FR LCD, which are also dedicated for biological indicator incubation. In 7 minutes, you can also um, use this device because it has a dedicated protein test position, only one, and 7 minutes. The quantification range is from 1 to 50 micrograms. Below 1, it will say less than 1 or 0, depending. And if it's between 0.5 and 1, or 0 and 0.5, okay? And beyond 50, it will only say more than, okay? But between 1 and 50, it will exactly give you um, the micrograms of protein in the sample that you have tested with the swab, okay? I did not talk about the reaction, but just quickly because we are running out of time and I still have a few slides more. But this reaction is modified burette reaction. Okay, we have a copper sulfate solution and BCA or bisinconinic acid solution. The peptide bonds of the primary structure of the proteins will react with the copper, will reduce the copper, and the copper reduced can bind to the BCA and form a purple complex. This is why the more dark the color, the darker the purple, more proteins I have in that solution. Okay? So which are the tips for a correct cleaning verification test by using a quantification protein test? Write clear and detailed instructions for use to ensure reproducible, standard operating procedures to make sure that we can reproduce the results. We need to compare. If we want to use a protein test, we have to be sure that we do every time we do the test, we do it in the same way. We have to standardize the frequency and respect it. We have to select only instruments that are highly challenging to the washing process, not the easy to clean one, the challenging ones. We have to perform always the swabbing in the same region and the same instruments. Okay, again, we want to compare. We want to make sure that we keep reducing the amount of proteins. If we don't follow a specific standard, like the ones I have shown as example on the tables, on the table, the previous table, at least I have to know where I'm standing, how much do I have, and try to reduce it as much as I can, preferably to 0. Okay, that's the idea. And of course, not only we have to define the instruments and swab every time the same, but also we have to choose which part of the instrument is a challenging one and only swab that surface because in the end is difficult to clean. Okay, and always perform the same swabbing. This way I can make comparisons. But what about our cannulated devices? We have been talking about surfaces. What about the internal channels of our cannulated devices? Are they truly clean? So let's take a look at the difficulties added to these devices. We all know what is a cannulated device. Just quickly, it's a medical device that contains a hollow channel or lumen inside. This channel allows the passage of liquids, air, or other instruments through them, are commonly used in surgical or diagnostic procedures, and are more difficult to clean and sterilize due to their hollow structure. Therefore, they require specific procedures to ensure that no residues remain inside the channels. I will take just 5 to 8 minutes more, okay? So, on the left side, we have the clinical frontline. We have artificial intelligence, telesurgery, sub-millimeter precision. But what do we have on the left, the right side? the reprocessing backline. We are still reprocessing our instruments in sinks, in manual sinks, and performing visual inspection. So complex endoscope or complex technology harbor hidden multi-drug resistant threats. The more complex the endoscope or the more complex the device for surgeries, the more probably or the more technology I need to make sure that I will correctly reprocess this instrument. So which are the stakes? Millions of procedures are performed annually, but reusable endoscopes present severe cleaning challenges due to their intricate designs. The threat is that numerous studies and independent organizations have linked endoscopes to outbreaks of multi-drug resistant microbes. And the reality is that in these outbreaks, visible residual soils is frequently discovered during these investigations, proving that standard decontamination steps often leave high concern organisms behind. So visual inspection is necessary, but entirely insufficient for safety. What the eye sees, what the borescope and chemistry see. Guidelines dictate that endoscopes must be visually inspected for cleanliness, missing parts, and damage prior to high-level disinfection or sterilization. The limitation is that the human eye cannot detect microscopic bioburden, cellular debris, or early-stage biofilm formation inside narrow 1 to 3 millimeter channels. While the tools like borescopes improve the detection of internal damage and retain gross soil, they cannot detect molecular level contamination that neutralizes the high-level disinfectants and sterilants. So for endoscope reprocessing, we also need to review what we are going to do to verify that we actually don't have organic residues behind our channels. And this is also why lately, same examples, the UK has always had these recommendations about protein tests, and this is related to prion outbreaks, okay? In the UK, the first prion investigations and scientific detection of these diseases were investigated, and this is why they have long time ago adopted protein tests for the correct verification of medical device reprocessing. But ISO and AME have also updated lately to recommend also protein tests for internal channels, specifically talking about endoscope or cannulated devices, as you can see here in these examples. So this is the product that we offer for internal channels. It is the same protein test that you have seen, is the same chemistry and the same associated technologies. You can see that you have the same devices for incubation and software, of course, for traceability. The little difference between them is the swab. Okay, we have on one hand the swab for internal channel monitoring or swabbing, and the protein test on the other hand, or the protein device detection for the chemical reaction to happen. So the swabs, we have 4 different options offering 4 different sizes for different internal diameters, all of them 2.5 meters long. The sizes are these: 1.7 to 2.7 and 3 millimeters. This is the size of the head of the swab. So, of course, with the swab, you will take the sample, and after you activate the pen, you put the swab and you incubate 4 minutes or 7 minutes in exactly the same way, because the reaction is exactly the same. Okay? Same devices, same technology, but a dedicated product for verification of protein residues inside the channels. So, in summary, and we are ending, protein quantification as a cleaning verification procedure will, in the end, assist in a continuous improvement strategy. Because if we define our protein baseline, we establish our protein thresholds, and we monitor our trend, we make sure that the lower protein levels over time reflect process optimization. Okay, so if you don't just say, okay, I will follow this or this guideline or this standard, you just make sure with just these simple steps that you are optimizing your process over time. And trend analysis will transform monitoring into risk management. In conclusion, don't just clean, monitor and verify. Patient safety and regulatory compliance rely entirely on an unbroken chain of parameters. From the first microscopic cavitation bubble, going through the rotation of a water spray, to the corroboration of protein residues, every variable of a cleaning process must be measured, tracked, and proven. Thank you. Okay, I will come back to you.
We have two questions, Anton.
Thanks.
Um,
Applauses.
Somebody asked
Yeah,
about how many indicators you put in one cycle. I think, I don't know,
I'm trying to just
one
one.
each level or rack every cycle. Maybe they are talking about splats.
Uh,
How many indicators you put in one cycle?
okay.
I'm assuming it's splats.
Yeah, I think so. So, um,
Or thermodisinfection, maybe.
it could be
Both.
for both of them because in
Exactly.
the end, we are just monitoring different parameters for the two steps of the process. But if we focus just on washing, our recommendation, or let's say a good practice, would be to monitor each level each time, because different levels rely on one rotating arm, and different things can happen with a rotating arm, one working and one not working. So in the end, monitoring all the levels I have would be a good practice. We just have to try to cover as much as possible with this type of simple indicators. What I like always when I work close to our to users or to our partners, I think a very good practice that will make very useful to have this indicator is to define zones. So I have levels and zones. So for example, let's say I have a wash disinfector with 3 levels and two different doors. Maybe it's a pass. If it's a pass-through washer, I will have 2 doors, right? So I can define levels and zones, like let's say A, B, C, D, right? Corners and middle, right? So every time I put an indicator, I have to make sure that I am using the defined zones and locations. This way, just by using this simple indicator, I can compare. I can compare between cycles. I can compare between loads. And changing location from cycle to cycle, respecting the defined zones, but changing locations. In the end, if I have 10 cycles per day, I will be monitoring the full chamber performance and all the situation that can happen regardless of the location of my instruments inside the chamber. Okay? So, answering specifically about this question, A good practice would be to do this. But if you use at least one indicator per cycle, changing location in defined zones, in the end, you will be during the day monitoring different parts and different situations inside that chamber. And same with the washer disinfector, the thermal disinfector indicator. I hope I have answered the question. Just let us know if it's clear.
Yes, I think it's clear, and
I'm not sure if there are more questions. Here,
Mm,
Javier, you're welcome, Javi.
yes, because Akash is just asking for a contact for an importer. We will
I'm not able
contact.
to see the same.
You don't see the chat box?
No, no, not the same ones. I mean the one that you have just read. I could not find it.
Akash
But
says require indicator in India. Can I get importer list? And
oh, okay,
so
uh,
I took note of that, and I
yeah,
will
yeah.
we
We
will
will
contact
contact
them with the
you.
distributor in India.
Yeah, yeah.
Um,
Javi,
yes,
and then Javi, I have a question about the ultrasonic color change indicator. Does the color change ever reverse? In other words, if it turns yellow, can it stay yellow for years, or will it eventually return to its original color? No, it actually reverse. Okay, the color is not stable, mm, because the reaction uh, only happens because the energy is provoking this chemical reaction, which is reversible. Okay, but in this case, once the energy delivered for the right amount of time, mm, for measuring the energy delivered to that indicator, will only remain for a few minutes, and then it can start to reverse back. This is the reason why, and this is one also other reason why we have developed this cavitation, the cavitest machine, the auto reader, so that you can immediately read and interpret the energy, the cavitation index through the precise measurement of the color, mm, without hesitation about is it uh, darker green, yellow, or would it be right? So to avoid these confusions, you will have immediately the result, instantly the result with the cavitation machine. Ideally, the incubator should be linked to a reporting system so that the indicator can be exactly. That's the main reason of using or having developed this auto reader, because it will allow you to digitalize the result, to have a printed evidence of the index of the cavitation index, which is in the end a translation of the energy delivered by that machine at that time to clean the posterior load that was going to be washed.
it's funny because Javier speaks Spanish and we are speaking with him in English, so it's weird, right?
He's the-
It's
he
a
and
special
yeah,
situation.
he's the exception of the
Yes,
meeting.
absolutely.
Thanks, Gabby, for joining. Yeah, our life. We basically spend all our working hours speaking in English. Is there any other questions? Don't be shy. And of course, if someone here does not want to make a question now, you can perfectly send us an email. We have the contact from your distributors, so you can contact them ask them any question or any an unanswered aspect about the products. There were too many to cover. I know I went too fast to cope up with the information. We will probably hang this webinar in our website, or make sure we can deliver the recording of the webinar to to be available for everyone. Okay. Um,
Toril is asking, will we get the presentation?
you're very welcome, Tariel. Let me know where are you from. Thanks for asking. Oh, oh, great. Thanks for joining us. Um, we can share the presentation with our partners. I'm not sure. I mean, we don't share the presentation, the PPT file, let's say, okay, to openly, but the recording, we will make sure that everyone attending this webinar will have access to the recording. Okay. But if you also have our contact, even through customer service, um, you can reach us, and we can get in contact to discuss about this. Okay.
Great.
I think there are other questions. Oh, I know now.
Anton?
Do you require or recommend splat test for ultrasonic washers, mainly used for pre-washed cycle? Cheers. Yes, yes. Regardless of how you use the ultrasonic, because in different markets or different situations, ultrasonic washers are used differently. There are some countries where they perform manual ultrasonic and then washer machine, but not all these steps are followed everywhere. So regardless of why you are using the ultrasonic machine, you have to make sure that you are actually cleaning the instruments. So the splat indicator is for load release, okay? The ampoule one, the CDWU, the cavitation test, is for making sure that the machine is working without the load. Once you put the load, you can use a splat test with the corresponding holder to make sure that you are correctly removing the organic residues by removing the test soil printed on the indicator. So yes, you can use the splat for load monitoring or load release in ultrasonic washes. I will share with you my screen again just to invite you to our next webinar, which will be in about 15 days. So next webinar will be on 20 and 21 of May. You should check your local time. We will start sending invitations. The title is The Hidden Gap in CSSD, from Data Collection to Real Insight. It will be delivered by Mr. Luciano De Marco, which is a commercial leader of digital solutions. So we will be presenting our BioNova Q software, which is not only a traceable solution, process traceability solution, because you can automatically digitalize and interpret the results from every step, as you have seen for washing indicators, protein tests, but also chemical indicators and biological indicators. So our system will allow you through our electronic devices to reach and automatically have all these results traced down by the process in the software. Besides this, it's also an instrument tracking system. So we have a perfect ecosystem where you will have the full workflow covered from the instrument perspective and the process efficiency perspective. Okay, so this is what we are going to present in this webinar. So be aware of our social media where we will be providing the local schedule details very soon. Okay? So if there are no further questions, please do not hesitate to contact us. You have our contacts uh, everywhere. You can find us on our website, uh, terragene.com. Very easy. You have a lot of contacts in there. You can contact your local representative or distributor that will reach us. Okay? You're muted, Caro.
Yeah, yeah, absolutely. They can contact us or our or their distributors if they
Okay,
have
so
any further
yeah,
questions.
thank you. Thank you also for the company. And thank you very much for connecting and listening to us. I hope this was interesting for everyone. And until next time, okay, we keep in touch. And thanks. Have a great rest of the day.